Hello everyone, my name is Vansika Padungsit, or you can call me Asanfa. If you have any question or any problem, you can contact me via this email, vansika_tu@hotmail.com. Okay, our lecture in the cell and molecular biology class today is the topic of enzymes. Let's start with the outline for my talk today. The topics for today was divided into 9 topics, including enzyme definition and characteristics, the structure of enzyme. Then we will talk about the topics of cofactor and coenzyme. After that, we will move to these 5 important topics, including mechanism of action, enzyme thermodynamics, enzyme kinetics, enzyme inhibitor, and regulatory enzyme. And the last topic for today, we will talk about the classification and nomenclature of the enzyme. Okay. The first topic for today is enzyme definition and characteristics. The first question when we talk about enzyme is, what is enzyme? So this is the definition of the enzyme. Enzyme is a substance produced by the living organism which acts as the catalyst to regulate the rate of the specific biochemical reaction. So after this, when the people ask you what is enzyme, so you can answer the definition of the enzyme like this. Enzyme, the substance produced by living organism which act as the catalyst and regulate the rate of the specific biochemical reaction inside the cell. Then the next thing that you have to know about enzyme is the important characteristic of enzyme. The first characteristic of enzyme is nearly all enzymes are protein. So what does it mean? It means not all enzymes are protein, but most of them are protein. And there are some other type of molecules that can act as enzyme also. And we will talk about this later in the next slide. The second characteristic of enzyme is the biological macromolecules. It's the last side of molecules. Enzyme accelerate the chemical reaction, or we can call that enzyme speed up or increase the rate of the reaction. The next thing, enzymes typically do not act alone. They need the helper molecules that we call cofactor and coenzyme to help them for their functions. Enzyme is highly specific catalyst. It means one type of enzyme catalyze their own specific biochemical reaction. One type of enzyme cannot speed up all type of the biochemical reaction, only the specific reaction that they can catalyze. The next thing, almost metabolic processes in the cell needs enzyme catalysis. Inside the cell, without the enzyme, the biochemical reaction can be occurred, but in the very slowly. So the cell needs enzyme to speed up the rate of the reaction. Then enzymes are not used up and remain unchanged at the end of the reaction. It means enzyme regulating the rate of chemical reaction without itself being altered in the process. So then in the end of the reaction, the enzyme remain unchanged and not used up. So there is the reusable molecules. And the last one, the very important characteristic of enzyme is enzyme increase the rate of the reaction by lowering or decrease the activation energy here. But enzyme not alter the equilibrium of the reaction. So this is the key for the enzyme study. Is increase the rate by lowering the activation energy and not alter the equilibrium of the reaction.
Okay, as I told you in the previous slide, most of enzymes are protein, except ribosome and deoxyribosome, which are RNA and DNA molecules, respectively. Ribosome and deoxyribosome are capable of catalyzing specific biochemical reactions similar to the action of protein enzymes. For example, this ribosome in the theta-hymena, the serrated protozoa here. These RNA molecules catalyze its own cleavage by cut itself out of pre-mRNA to produce the mature RNA. The red color acts as the substrate, whereas the blue color, the alpha helix blue color here, acts as the active site of the enzyme. So this ribosome can cut the red color out from the pre-mRNA molecules to produce the mature mRNA. As you can see here by RNA molecules itself without any help from protein enzyme, it can catalyze its own cleavage, so we can categorize them as an enzyme. And for deoxyribosome, there are no known naturally occurring deoxyribosome. Most known deoxyribosome discovered through the in vitro study. For example, this 1023 deoxyribosome molecules here. This deoxyribosome creep between A and U at the RNA monomer within the oligonucleotide. So these DNA molecules act as an enzyme to cut the bond between A and U by using the RNA as a substrate. Okay, so now we know both ribosome and deoxyribosome already. Then next, we will have a look at the structure of enzyme. Enzymes are biological macromolecules. In general, enzymes are globular protein, can acting alone or in the larger complex. For example, amylase, which catalyze the hydrolysis of starch into the small carbohydrate molecule. The structure of amylase shown in this figure. As you can see, amylase is a single polypeptide chain. They consist of only the single polypeptide chain. So this enzyme can acting alone. Whereas most of enzymes are multiple subunits. For example, the neuraminidase, which is glycoside hydrolase enzyme that cleave the glycosidic linkage of the neuraminic acid. This figure shows the structure of neuraminidase. As you can see, neuraminidase exists as the large complex, or we can call this the multiple subunit enzyme. Neuraminidase consists of 4 identical subunits like this. So this is the structure of neuraminidase, which is the larger complex enzyme. From this figure, you can see that enzymes are made up of amino acids, which are linked together via the peptide bond in a linear chain like this. The primary structure of enzyme is the amino acid chain here. Then hydrogen in the amino group and the oxygen in the carboxyl group of each amino acid can bond with each other via the hydrogen bond. So the protein chain can flow up, resulting in the secondary structure of enzyme here, which can be both alpha helix like this, or it can be the beta strand sheet also. Next step, the protein can flow up further as a three-dimensional structure, which is the tertiary structure of protein. And then each subunit of tertiary structure, here and here, can bind together to form the quaternary structure, like this. If you remember from the previous slide, structure of amylase is the tertiary structure like this. They consist of only single polypeptide chains, whereas the structure of neuraminidase is the quaternary structure because they consist of more than one polypeptide chains. From this figure, we can conclude that the sequence of amino acid here specify the structure of enzyme which in turn determine the catalytic activity of the enzyme. We can conclude that sequence of amino acids specifies structure of enzyme which in turn determine the catalytic activity of enzyme. And when we have a look inside the structure of enzyme, it contains the region that we call active site. Active site is the region of an enzyme where substrate molecules bind and undergo the chemical reaction. The active site is small when compared with the whole molecules of enzyme. It usually consists of 3 or 4 amino acids, while the other amino acids within the protein are required to maintain the protein tertiary structure of enzyme. For example, this is the structure of lysosome. Lysosome consists of 129 amino acids, but only these 4 amino acids, 1, 2, 3, 4. Only these 4 amino acids are used for forming the enzyme active site. It is not necessary that only the adjacent amino acid can form the active site. Amino acid chain can form and form the bond between these 4 amino acids, which is not the adjacent amino acid to form the enzyme active site. The active site consists of 2 parts. The first part we call the binding site, and the second part of active site is the catalytic site. Binding site is the residue that form temporary bond with the substrate, whereas the catalytic site is the residue that catalyze the reaction of substrate. So this is the active site of enzyme, which consists of the binding site and catalytic site. Binding site used for the binding of enzyme to the substrate, whereas the catalytic site is the place where reaction occurs. From the previous slide, so now we know enzymes have an active site to bind to substrate and undergo the chemical reaction. So then the next question is how enzyme and substrate bind together. There are two models for enzyme substrate binding. The first one we call lock and key model. For lock and key model, at first, enzyme and substrate are complementary so that they fit together like lock and key and can bind together like this. The second model for enzyme substrate binding is induced fit model. For induced fit model, at first, enzyme and substrate are not complementary. But at the transition state, enzyme can change their conformation and then can bind to the substrate like this at the transition state. So this is the difference between these two models. For lock and key, at first, enzyme and substrate are complementary like lock and key, so then they can bind together. Whereas for the induced fit model, at first, enzyme is not complementary to substrate, but enzyme can change their conformation and bind to substrate at the transition state. Okay, let's see the lock and key model first. The lock and key model theory first postulated by Emil Fischer in 1894. This model shows the high specificity of enzymes. This model for enzyme substrate interaction suggesting that the enzyme and substrate processes the specific complementary geometric shape that fit exactly into one another, like the key into the lock. Only the correct size and shape of the substrate or the key here would fit into the active site or the keyhole of the lock or of the enzyme here. So, or we can call that the substrate would fit into the active site of the enzyme, like the key would fit into the keyhole of the lock. However, this model lock and key hypothesis can be misleading when applied to the enzymatic catalysis. An enzyme completely complementary to its substrate would be a very poor enzyme. And we will discuss this point later, why we can say that enzyme that complementary to its substrate would be the very poor enzyme. For the induced fit model, this model suggested by Daniel Kochland in 1958. This model is more accepted for enzyme substrate complex than the lock and key model. Induced fit model suggests that the active site of enzyme here continue to change until the substrate is completely bound to the enzyme. This active site at first is not complementary to the substrate, but is continue change until it can bind completely with the substrate. Induced fit models show that enzymes are rather flexible structure in which the active site of enzyme continually reshaped by its interaction with the substrate until the substrate is completely bound to the active site of enzyme. When enzyme and substrate bind completely, we can call this reaction transition state. Okay, this is the meaning of induced fit model. Okay, let's have a look at figure A first. Let's imagine this is the biological reaction. The substrate for this reaction we call the metal stick here. Without enzyme, this reaction can be occur in the slow rate. Before the stick is broken, it must be bent. Like this. At this point, we call it the transition state, when the stick bends before it's broken. And then finally, without the enzyme, we can also got the product as a broken stick here. Then we will have a look at figure B. The blue color here is an imaginary enzyme we call stick case. Stick case was designed to catalyze the breakdown of the metal stick. In figure B, as you can see, the stick case with complementary in structure to the stick stabilize the substrate like this. When it's complementary, then enzyme and substrate will bind together and stabilize the substrate like this. So the breakdown of metal stick is very hard to occur. We cannot get the product if the enzyme is complementary to the substrate. This figure is the example for the lock and key model, which at first enzyme and substrate are complementary, so we cannot get the product from the enzyme that complementary to the substrate at first. So this is the reason why we can say that an enzyme that complementary to the substrate would be the very poor enzyme. Whereas for the induced fit model in the figure C here, an enzyme will have the active site like this. The active site of this enzyme complementary to the reaction transition state is not complementary with the substrate but complementary to the transition state. This reaction helps to destabilize the stick, so the reaction is easier to occur. So this model is more accepted for the enzyme substrate binding than the lock and key model. Okay, so now we move to the next topic. As I told you in the earlier, enzymes are typically do not act alone. Enzymes need helper molecules that we call cofactors and coenzymes. Most of enzymes require the helper molecules, but not all. For example, lysosome. Lysosome is protein-only enzyme, so no need any helper molecules for their function. But most of enzymes require the additional chemical component for their function. There are two types of helper molecules, cofactor and coenzyme. Cofactor is an inorganic ion, or we can call it a metallic ion such as ferrous or ferric ion, manganese, magnesium, or zinc ion. Whereas the coenzyme is the metallo-organic molecules. It means it contains both part of metallic and part of organic molecules. Almost all coenzymes are derived from the vitamins such as NAD+, NADP+, or the coenzyme A. Cofactor and coenzyme are required for the enzyme biological activity and can be considered as the helper molecules that assist the biochemical transformation. For the topic of cofactors and coenzyme, there are three terms that you should know in this slide. The first one is post-titic group. Post-titic group is mean the coenzyme that tightly bound to the enzyme. So we call this coenzyme as post-titic group. The second term is apoenzyme. Apoenzyme is mean the inactive enzyme without the cofactor and coenzyme. So the enzyme alone without cofactor and coenzyme in the inactive form we call apoenzyme. And the third term in this slide that you should know is holoenzyme. Holoenzyme is the whole enzyme, or we can call the complete enzyme with cofactor and coenzyme. This holoenzyme is in the active form. Okay, this is three terms that you should know about cofactor and coenzyme. One of characteristic of enzyme is enzymes are highly specific catalysts. Enzymes show two types of specificity. The first one is relative specificity and the second one is absolute specificity. For the relative specific, this type of specificity, the enzyme would bind to another type of substrate if that substrate resembles the complementary one of enzyme. For example, the alpha amylase. Alpha amylase specific to alpha-1,4-glycosidic bond. As you can see from this figure, both amylose and amylopectin contain the alpha-1,4-glycosidic bond. So this alpha amylase can be catalyzing both amylose and amylopectin which contain alpha-1,4-glycosidic bond. This is the meaning of relative specific. For absolute specificity, the enzyme would only bind to one particular type of substrate and no other. For example, lactase enzyme. Lactase can hydrolyze beta-1,4-glycosidic bond of lactose only. So, lactose is the only one substrate of lactase. Even the cellulose also contains this beta-1,4-glycosidic bond, but lactase cannot catalyze the reaction for cellulose. So this is the meaning of absolute specificity. Then let's have a look why almost metabolic processes in the cell need enzyme catalysis. When we have a look at this table, you can see that without enzymes, this reaction takes place at very slow rate and make life impossible. For example, the protein hydrolysis reaction. Protein hydrolysis takes 400 years at 25 degrees Celsius. And when we increase the temperature to 100 degrees Celsius, the reaction rate of protein hydrolysis is increased. It will take around 5.5 weeks without enzyme. But in real life, the temperature in the body is not more than 27 degrees Celsius, so we cannot increase the rate of the reaction by increase the temperature. So we need the enzyme to speed up the reaction rate and make the life possible. The rate enhancement of enzyme is rank of 10 to the 5 to 10 to 17. So this enhancement rate is make life possible. This is the reason why metabolic processes in the cell needs enzyme catalysis. Then next, enzymes are not used up and remain unchanged at the end of the reaction. Enzymes are not used up and are not changed by the reaction. Enzymes are used only temporarily, and after the end of the reaction, when we cut the product from the reaction, the enzyme molecules here can be used again for the same reaction. So we can call that enzymes are reusable molecules. And we need only the little amount of enzyme for the reaction. Then now we move to the important topic for the enzyme, mechanisms of action of enzyme. Enzymes speed up the chemical reaction by increase the rate of the reaction. Okay, first, let's have a look on the simple enzymatic reaction shown here. E is represent the enzyme, S substrate, P products. ES and EP are transient complex of enzyme with the substrate and product respectively. At first step, free enzyme here and free substrate form the transient complex ES. Then change to transient complex of EP, enzyme and product. And in the last step, enzyme release from the product. As you can see, every step during the enzymatic reaction is reversible. This is reaction coordinate diagram with our enzyme, which plot between the free energy in the y-axis and reaction coordinate at the x-axis. When we have a look at this reaction coordinate diagram, there is an energy barrier between substrate and product. And to undergo this reaction, the molecules must overcome this barrier and must raise to the higher energy level here. At the top of energy hill is called the transition state. The starting point of either forward reaction or reverse reaction we call the ground state. This is the ground state of substrate for the forward reaction, and this is the ground state of product for the reverse reaction. Then the activation energy is the energy required to start the reaction, which is the difference between the energy level of the ground state and transition state. So in this figure, we have two values of the activation energy from the forward reaction from substrate to product. The activation energy defined by this level. Whereas from the reverse reaction from the product to the substrate, the activation energy defined by this level. Okay, in conclusion for reaction coordinate diagram, this diagram plot between free energy and reaction coordinate. The term that you should know in this curve is the ground state, which is the starting point for either forward or reverse reaction. The transition state, which is the higher energy or the top of the hill of the energy. And the activation energy, which is the difference between energy level of ground state and transition state. It can be the activation energy for the forward reaction here, or the activation energy in the reward reaction here. Activation energy have the abbreviation, we can call it delta G plus plus or the EA here. This is reaction coordinate diagram without enzyme. And then let's have a look at the reaction coordinate diagram in the presence of enzyme in the blue line here. What is happened? Only one thing was changed. The reaction that catalyzed by enzyme show the lower activation energy. The amount of activation energy with enzyme is less than without enzyme. As you can see, the delta G cat here is less than delta G uncat here. So we can conclude that enzymes speed up the rate of the reaction by lowering the activation energy. This is the key for the enzyme to speed up the reaction. They speed up by lowering the activation energy, and you can see in the coordinate diagram already, delta G cat is less than delta G uncat. Okay, did you remember this figure for lock and key and the induced fit model? So now we will have a look at the reaction coordinate diagram of each reaction. In figure A without enzyme, the activation energy or the delta G plus plus is equal to this amount. Then in the figure B, for the lock and key model, when the enzyme complementary to the substrate, what happened? The ES complex is more stable and has the lower free energy in the ground state than the substrate alone. Result in the activation energy is increased. This is the activation energy of the enzyme that complementary to the substrate. When compared with the reaction without enzyme here, as you can see, delta G cat is greater than delta G uncat. Then I will introduce you the next term that we call the delta GM. Delta GM is the difference between the transition state energy of cat and uncat. Or you can calculate by delta GM is equal to delta G cat subtract by delta G uncat. For figure B here, delta G cat is greater than delta G uncat, so this delta GM have the positive sign. And this coordinate diagram show that the rate of the reaction will be decreased. Okay. Then in the figure C for the induced fit model, the enzyme complementary to the transition state. So this ES complex still undergo the increase in free energy, and this complex is not stable. In the same time, enzyme lowering the activation energy. Here, delta G cat of this reaction that enzyme complementary to the transition state is less than the delta G uncat here. Then the delta GM have the negative sign. So from this coordinate diagram, we can conclude that the rate of the reaction will be increased. Okay. Again, now we know the enzyme are speed up the reaction by the lowering the activation energy. The next question is how enzyme lowers the activation energy. What do enzyme do to lower the activation energy? The answer is there are two types of interaction involved. The first interaction is the rearrangement of covalent bond during the enzyme catalyzed reaction. And the second type of interaction that involved is the non-covalent interaction between enzyme and substrate. One way that enzyme used for lowering the activation energy is the interaction of rearrangement of covalent bond during the enzyme catalyzed reaction. Catalytic function group on enzyme may form the transient covalent bond with the substrate, or sometimes a group may be transient transfer from the substrate to the enzyme. These covalent interactions between enzyme and substrate lower the activation energy and speed up the reaction by providing the alternative lower energy reaction path, as you can see in the coordinate diagram with the enzyme. The other interaction that enzyme use for the lowering the activation energy is the non-covalent interactions between enzymes and substrate. Much of energy required to lower the activation energy are derived from this weak non-covalent interaction between substrate and enzyme. We can call it come from the formation of specific ES complex. The interaction between substrate and enzyme, including hydrogen bonds, hydrophobic and ionic interactions. Formation of each weak interaction in the ES complex is accompanied by the release of the small amount of free energy. This energy derived from enzyme-substrate interaction is called binding energy or delta GB. The binding energy is the major source of free energy used by enzyme to lower the activation energy of the reaction. So this is two way or two interaction that enzyme use for lower the activation energy. The first one is the covalent bond and the second one is the non-covalent bond, which is the major source, and the energy that released from this non-covalent bond we call it binding energy or delta GB. In conclusion, enzyme increase the rate of the reaction by lowering activation energy, and there are two sources of energy used for lowering activation energy. The first one is from the transient covalent bond between functional group of enzyme and substrate. And the second one is the major source that we call the binding energy or delta GB here, that generated from the non-covalent bond between enzyme and substrate. Okay, so now we will move to the next topic, enzyme thermodynamics. When we talk about enzyme thermodynamics, we talk about the equilibrium of enzyme. As I mentioned in the beginning of this lecture, enzymes just increase the rate of the reaction, but it not alter the equilibrium of the reaction. And the equilibrium of enzyme involved with the term of the standard give free energy change or the standard free energy chain that have a sign like this, delta G naught. Delta G naught equal to the free energy at the ground state of product subtract by standard free energy at ground state of substrate. Delta G naught here equal to standard free energy of product here subtract by free energy of substrate. So from this figure, you can see that delta G naught should be have the negative sign because the free energy of product is less than free energy of substrate. When we have a look at reaction coordinate diagram without enzyme here and with enzyme here, you can see that with or without enzyme, the standard free energy change or delta G naught here and here still the same. That means enzyme not alter the equilibrium of the reaction. If free energy change came from ground state of product subtract by ground state of substrate, so the free energy change can either be negative or positive sign. In the reaction with free energy change is negative, or when the ground state of product is less than ground state of substrate. This reaction is spontaneous reaction. They can occur by themselves, or we can call this reaction exergonic reaction. Whereas the reaction with free energy change is positive. Here, when the ground state of product greater than ground state of substrate. This reaction is not spontaneous. This reaction needs the energy from outside to drive the reaction, or we can call endergonic reaction. But if the free energy change is equal to 0, that means the reaction has achieved equilibrium. The rate of forward reaction will equal to the rate of reward reaction. But notice for the exergonic reaction here, the spontaneous reaction is not mean the rate of reaction is high. For example, if you remember the protein hydrolysis reaction, that reaction is a spontaneous reaction, but it takes around 400 years without enzyme. Okay. We can calculate the free energy change from the thermodynamic equation here. The delta G naught is equal to minus RT ln KEQ, where R is gas constant is equal to 8.3 something, and T is the absolute temperature at 298 Kelvin. And KEQ is the equilibrium constant. KEQ is equal to concentration of product divided by concentration of substrate here. And when we have a look here, when the concentration of product is equal to concentration of substrate, then the KEQ is equal to 1. And when we have a look in this term, ln KEQ. If KEQ is equal to 1, ln 1 is equal to 0 if you remember. You can use your calculator to calculate ln 1 is equal to 0. Then from this, the term of delta G naught or the free energy change should be 0. At this point, the reaction has achieved the equilibrium like this, because delta G is also equal to ground state of product divided by ground state of substrate. If this delta G is equal to 0, so this reaction is achieved the equilibrium. Okay, this is one way to calculate the free energy change by using the thermodynamic equation. Okay, let's have a look at the thermodynamic equation again. When the concentration of the product is greater than concentration of substrate, when P greater than S, so the KEQ is greater than 1, right? And when KEQ greater than 1, learn something that more than 1 have the positive sign. And then for all, we have the negative side here. For all, the delta G naught should be have the negative sign in this case. As you can see here, delta G naught less than 0. So you can draw the coordination diagram like this. When the concentration of product greater than concentration of substrate, this reaction should be exergonic reaction. The other way around, when the concentration of product is less than concentration of substrate, then KEQ in this case, it should be less than 1, right? Then learn something that less than 1 should have the negative side. Then we have negative here. So for all in this case, the delta G naught should have the positive sign. Or you can see that the delta G naught is greater than 0 here. Then you can draw the coordination diagram like this. And this is the endergonic reaction when the concentration of product is less than concentration of substrate. You should notice that for the coordination diagram, the y-axis as the gift free energy, not the concentration. So please aware this.